I'm Dr. Jim Zoval. You're about to watch one of my video lectures. If you like this video and would like to get videos for your entire General Organic and Biochemistry course, you can do so at www.zovallearning.com. I'll give you more information about that at the end of this video. The energy that's contained in food can be traced back to the sun. These videos are made to be viewed while using fill-in lecture notes. The pause in the audio is to give students time to complete the lecture notes fill-in. The lecture notes can be downloaded, free of charge, at www.zovallearning.com. Energy from sunlight is captured by plants during photosynthesis as they convert CO2 and H2O into glucose and oxygen gas. Without the energy from sunlight, the reaction of CO2 with H2O producing glucose and oxygen gas is not spontaneous. You could test this yourself by exhaling CO2 through a straw and into water. If glucose were produced spontaneously, then eventually you'd be able to taste the sweetness of glucose in the water. If you did this, though, you would find that the solution does not taste sweet. The reaction does not occur on its own because glucose and O2 gas are higher in free energy than our CO2 and H2O. This reaction can only become spontaneous with the input of energy from sunlight. In photosynthesis, energy from the sun's light does not vanish. It is converted to chemical potential energy within glucose. Plants store excess glucose as starch. One way to extract the energy from starch is to burn it. In fact, granaries, which are factories that extract and store starch in the form of flour, are very dangerous because of the aerosolized starch. Huge explosions and fires have occurred in granaries when one spark ignites the aerosolized starch particles. In such fires and explosions, the energy is wasted. None of it's used to do work. If the starch were burned under a sealed container that has some water in it, the energy released from the starch would cause the water to boil, and the resulting high-pressure vapor could be used to move a piston. This is how steam engines work, although fuels other than starch are used in steam engines. Humans did not evolve steam engines as power supplies. Instead, we use a series of catabolic chemical reactions to slowly oxidize food, eventually converting it back to CO2 and H2O. Energy that's released in these oxidations is converted to chemical potential energy within ATP. Not all of the energy released by the oxidation of food is converted to energy within ATP. Some is lost as heat. Organisms lose about 50% of food's energy as heat. It is this heat that allows us to maintain our normal body temperatures in cooler environments. Note how nature cycles glucose and O2 with CO2 and H2O using photosynthesis, an anabolic process, and glucose catabolism. All three classes of macronutrients in food, carbohydrates, triglycerides, and proteins, are catabolized in 4 stages: digestion, acetyl-coenzyme A production, the citric acid cycle, and oxidative phosphorylation. For those of you who are following this video using the fill-in lecture notes, click pause and complete these fill-ins. Stage 1. The digestion of carbohydrates. Digestion is the process in which the body breaks down carbohydrate, protein, and triglyceride polymers into their monomer residues. For example, carbohydrate polymers are converted to monosaccharides. Digestion occurs in the digestive system. The digestive system, sometimes referred to as the digestive tract or the gastrointestinal tract, includes the organs that are responsible for digesting food and eliminating the undigestible components of food. The major organs of the human digestive system are shown in this view graph. During the digestion of carbohydrate polymers, most oligosaccharides and polysaccharides can be broken down to monosaccharides. These reactions are catalyzed by digestive enzymes. Approximately 50% of our dietary carbohydrates are in the form of starch. In a previous chapter, you learned that starch has two components, amylose and amylopectin, both of which are composed entirely of glucose residues. Digestion of amylose and amylopectin, starch, begins in the mouth. Saliva contains salivary amylase enzymes, which catalyze the hydrolysis of some of the alpha 1,4 glycosidic bonds in amylose and amylopectin. In the hydrolysis of carbohydrates, water molecules are used to lyse, that's to break, glycosidic bonds. The hydrolysis of a glycosidic bond is the reverse of the reaction that you learned about in the chapter on carbohydrates for the formation of glycosidic bonds. An example of the hydrolysis of one of amylose's alpha 1,4 glycosidic bonds is shown in this view graph. You're looking at an illustrative overview of the digestion of carbohydrates. Digestion of carbohydrates begins in the mouth, where salivary amylase catalyzes the hydrolysis of amylose and amylopectin to form maltose, which is an alpha 1,4 glucose-glucose disaccharide, and small oligosaccharides called dextrins. Dextrins are oligosaccharides that generally contain between 3 and 8 glucose residues. The maltose and dextrins, along with other non-starch dietary carbohydrates, then pass through the stomach. Carbohydrate digestion temporarily stops in the stomach because the salivary amylase enzyme is denatured by the stomach's very low pH environment. Digestion continues in the small intestine with the help of more digestive enzymes. Pancreatic amylase enzymes catalyze the hydrolysis of dextrins to form maltose and isomaltose. Maltose is a alpha 1,4 glucose-glucose disaccharide. Isomaltose is an alpha 1,6 glucose-glucose disaccharide. The isomaltose comes from the alpha 1,6 branching points that were originally in amylopectin and remain in some of the dextrins. Maltase and isomaltase enzymes catalyze the hydrolysis of maltose and isomaltose, respectively, into glucose. The non-starch dietary carbohydrates, lactose and sucrose, are converted to monosaccharides with the help of lactase and sucrase enzymes. Lactose is hydrolyzed to galactose and glucose. Sucrose is hydrolyzed to fructose and glucose. It's critical that oligosaccharides and polysaccharides be converted to monosaccharides in order for the sugars to pass through the intestine wall and into the bloodstream so that they are available to cells throughout the body. Monosaccharides are transported into cells by passive diffusion through transmembrane proteins. Not all dietary carbohydrates can be digested. For example, cellulose can't be digested by humans because we don't have the enzyme that's capable of breaking a glucose-glucose beta 1,4 glycosidic bond. Cellulose cannot pass through the wall of the small intestine and therefore passes through the digestive tract until it is excreted in feces. In this video, I introduced you to the metabolism of carbohydrates. I listed the 4 stages in which the macronutrients in food are catabolized. Then I elaborated on digestion, the first stage of carbohydrate catabolism. In the digestion of carbohydrates, carbohydrate polysaccharides and oligosaccharides are converted to glucose and some other monosaccharides. In the next video, I'll discuss stage 2 of carbohydrate catabolism. You can get your entire general, organic, and biochemistry course as a series of video lectures. The rental period is for 1 year, so you'll have these video lectures for the entire term of the course. You could watch the videos whenever and however many times you wish. I survey my students, and they prefer these video lectures over the textbook by a ratio of about 10 to 1. Fill-in lecture notes and an electronic version of my textbook are available free of charge at the website. To get more information or to get the videos, free textbook, and free lecture notes, visit my website at www.zovallearning.com.